So let's review kind of what we went over last time. So remember the whole thing that we were covering last time ended in what's called the nephron. And the nephron, remember, is the structural and functional unit of the kidney. So it's where everything that the kidney does, this is where it happens. And remember, the kidney is all about blood. It's all about taking the blood and cleaning it, removing all the waste products, and then adjusting things like the volume, the composition, all that stuff. And so it all happens here at the nephron. And we went over the structure of the nephron. Remember, there are three parts to the nephron. There's a part that involves blood vessels. And so that starts here with the afferent arteriole. And then we have the glomerular capillaries. And then we have the efferent arteriole. And then we have the peritubular capillaries and the vasa recta. So that's the vascular portion of the nephron. But then remember, we also have this long tube. And that starts here with Bowman's capsule. And then it goes from there to the proximal convoluted tubule, and then proximal convoluted tubule down to the loop of Henle, which has a descending and ascending limb, and then the distal convoluted tubule, and then finally the collecting duct. So that's the tubular portion of the nephron. And then the last thing we talked about was right here where the afferent arteriole and the distal convoluted tubule touch each other is a special part. And that part regulates the flow through this nephron. And remember, if you look at the afferent arteriole, we have these cells called JG cells. And if you look at the distal convoluted tubule, we have these special cells which are called macula densa cells. So if you put those together, those are the regulatory portion of this nephron. So there's a vascular portion, a tubular portion, and a regulatory portion. Another thing we said was that there are two types of nephrons in the human kidney. There are these which have a short loop of Henle. They're called cortical nephrons. And remember, about 80 to 85% of human nephrons are cortical. And then here, this one, much longer loop of Henle. These are called juxtamedullary. And so these are many fewer. So only about 15% are going to be juxtamedullary. So that's the nephron in kind of a review. Anybody have any questions about any of that? Everybody remember it okay? So what's going to happen is blood is going to come through here and it's going to be filtered, it's going to be cleaned, it's going to be refreshed, it's going to be adjusted, and then it's going to go back into the general circulation. And remember, this happens about 60 times a day. It happens about 60 times a day. So 60 times a day, your entire liquid portion of your blood is filtered out. Well, if we think about the liquid portion of your blood, remember we have about 5 liters of blood, but about 55% of it is the plasma, the liquid portion. Remember we did this little centrifuge thing. This is about 55%. That's about 3 liters of fluid. Well, if you filter 3 liters of fluid 60 times a day, that means that we're going to do and create 180 liters that's going to be filtered. Well, anything that's filtered is called filtrate. So we're going to produce about 180 liters of filtrate. And then what's going to happen is that 180 liters is going to go through this whole long tube. And as it goes through the tube, we're going to take stuff out. We're going to put stuff in. And then by the time it goes through the whole tube, it's going to be urine. So filtrate is what comes in, and urine is what's going to go out. But there's 180 liters of filtrate. We don't have anywhere near that much urine. Think about it. If you urinated 180 liters, that's about 40 gallons, 45 gallons, 45 gallons of urine. Well, if you urinated 45 gallons, you'd also have to drink 45 gallons of water. So all you'd have time to do would be to drink water and go to the bathroom. So obviously, not all of this can go through and become this, because we only excrete about 1.5 liters a day, not 45 gallons. So in other words, it's right around 1%, a little bit less than 1%. So we're going to have to pull out a ton of water and a ton of other things when we make urine. And so it happens in 3 steps. And the 3 steps are filtration, reabsorption, and secretion. So what are those 3 steps? Filtration, we're going to filter the blood. We're going to filter it, and we're going to form filtrate. And remember, we're going to make about 180 liters of filtrate every day. Well, again, it's obvious if we make 180 liters here, and there's only 1.5 liters over here, that we're going to have to pull out a ton of liquid, a ton of other stuff. That's reabsorption. So reabsorption is we're going to reabsorb the stuff we want to keep. So reabsorb the valuable stuff. And then there's another process called secretion. And secretion is we're actually going to put stuff in here. We're going to put it in there, and it's going to wind up in the urine. So that's secretion. Secretion means that we're going to actively get rid of unwanted substances. We're going to get rid of extra stuff. So 3 processes: filtration, reabsorption, and secretion. So let's look at these 3 processes. And we're going to look at filtration first. Now this is a little movie, and you can watch it if you want to. You can't watch it on here though, but you can with the PowerPoint. So look, here are 3 processes. So again, filtration. Well, filtration happens right here. It happens at the glomerular capillaries in Bowman's capsule. That's where filtration happens. And so we're going to force fluid and dissolve substances out of the blood into this tube. That's filtration. But what comes out, some of this stuff is good stuff. It could be stuff like glucose, or it could be amino acids or something. Well, if it stays in here, it's going to be part of the urine, and we're going to lose it. So what's going to happen is when that glucose comes in here, or those amino acids come in here, we're going to pull it out. We're going to keep it. We're going to take it out of here and put it back into our bloodstream. And that is reabsorption. And then finally, remember there's stuff that we don't like enough that we just want to get rid of it. And so what we're going to do is we're going to take it out of our blood and put it directly into the tube. And if you put it into the tube, it's going to become part of the urine. And so that is secretion. So let's look at these. Let's look at filtration first. Well, in order to do filtration, you actually need three things. And I bet a bunch of you did filtration already today. How many of you made coffee this morning, or tea, or something like that? Well, when you do that, you do filtration. And so there are lots of different ways to do it, but in order to do filtration, you need three things. So you need a liquid. Well, when you make coffee, that liquid is water, but in our case, the liquid is blood. And you need a force to make it go where you want it to go. When you make coffee, it might be gravity or it might be a mechanical pump of some sort. Well, in our case, the force is blood pressure. And then the third thing you need is you need a filter. Well, when you make coffee, the filter could be a paper filter, it could be a metal filter, could be something like that. But here, when we look at the filter is those glomerular capillaries in Bowman's capsule. And so that's what we're going to see in just a second. So, three things. So look, if I want to filter this, I need three things. So here's my liquid, there it is. There's my filter, there it is. And then the force is gravity. And look, this is what's going to happen. So look, what I've done is I'm going to separate stuff. And I'm going to separate it as I pass it through this filter. Well, what makes a filter a filter? Anybody know? If you look at a coffee filter, how is it different from anything else? What's different? What makes a filter a filter? If you look at a metal filter, what's it got that's different? Anybody know? A filter has holes in it. So if I look at a filter like this one right here, there are little tiny holes in it. So look, the liquid went through, but this brown stuff, the residue, did not. Why doesn't that go through? Why isn't it here? Why doesn't it go through the filter? When you make coffee, you put coffee, ground up coffee in here. How come it doesn't wind up down here in the liquid? Yeah, it's all about size. That's exactly what it is. It's all about size, and that's the only way a filter works. So look, if I were going to make coffee, and here, maybe just like this, I'm going to do it just like this. Well, normally I put these grounds in here, and then I'm going to pour hot water through it, basically. If I put salt in the water, in other words, it's salty water, what would my coffee taste like? Is it going to taste like normal coffee? It's going to taste like what? It's going to taste like salt, because salt's going to go right on through and it's going to be in the coffee. What if I were to put sugar water here? What's the coffee going to taste like? It's going to taste sweet. In other words, the filter lets anything small enough to go through the holes it just lets it go through. The only regulation about this is on size. So let's think about blood. Blood, if you look at whole blood, it's got red blood cells, it's got white blood cells, and it's got little platelets in it, but there's tons of liquid in here. But remember, plasma is not just water. Plasma's got all kinds of stuff in it. It's got proteins in it, it's got sugars in it, it's got all kinds of electrolytes in it. And if I look at this, anything that's small enough to go through that filter is going to wind up in the filtrate. It's just based on size, nothing else. Well, if I look at this filter, look, it's just one layer of paper, one layer. But if I wanted to, could I add another layer of paper? Sure, I could. I could add 2 layers of paper. If I wanted to, I could do 3 layers of paper. Does the holes in the 3 layers have to be the same size in order for this to work? They don't. I could have big holes here, and medium-sized holes here, and little holes here. But what would get through? What would make it through? If this were big holes, and this were medium-sized holes, and this were small holes, who's going to regulate what gets through? The smallest, exactly. But look, it's no longer one layer, it's 3 layers. It's not a 1-stage filter, it's a 3-stage filter. And that's exactly what the filter on the kidney looks like. It's a 3-stage filter. The 3 stages are, first there's capillaries. Remember, some capillaries are fenestrated. Everybody remember fenestrated capillaries? Remember, they have pores or fenestrations. And remember, it looks like a colander or a strainer that you might have in your kitchen. So there's the largest holes. And then remember, capillaries have a basement membrane. Well, they have holes in them too, but they're medium-sized holes. But then don't forget, wrapped around the capillaries in the kidney, we have these big cells that have these feet. Remember, they're called podocytes. And those podocytes, remember, they wrap around, and another one comes and wraps like this. And they interdigitate. And what they create are filtration slits. And these are the smallest holes. So if we look at it, here's our capillary. And those are the biggest holes right there. There's the basement membrane, and it has medium-sized holes. And then here are those filtration slits. And they're the smallest holes. Well, this is how big the smallest holes are. They're about 3 nanometers, which means that anything smaller than 3 nanometers can pass through, but anything bigger cannot. So if we look at it, here's again what it looks like. Here's the capillary walls with holes in it. Remember, those are fenestrations. And then there's our basement membrane. And then here's that podocyte. And remember, they have feet. The feet are called pedicels. And remember, those pedicels interdigitate. So we have filtration slits. And so the blood, which is in here, gets pushed against the wall. And some things which are small enough will go through all 3 layers, and they're going to wind up as filtrate. So look, again, here's blood right here, and here's our 3 layers. There's the capillary layer right there. And see the pores? Those are those fenestrations. And there's the basement membrane, and then here's our podocyte, and here's our pedicels, and here's our filtration slits. And so blood is in here, and it's going to get pushed against the wall by blood pressure. And anything small enough to go through this and this and this, this and this and this is going to get pushed through, and it's going to be called filtrate. Does it make sense? Does everybody understand this filtration membrane, how it's not 1 layer but 3 layers? And the holes get smaller and smaller and smaller. That the capillary has the biggest holes. This is the biggest, and then we have medium-sized holes, and then the smallest holes right here. And anything small enough to go through the smallest holes is going to become filtrate. So that means anything in blood smaller than the smallest holes are going to become filtrate. That doesn't tell you if it's good or bad. In fact, we have some good things and some bad things. It's only separated based on size, not anything else. Not whether it's good or bad or whether we want to keep it or get rid of it. It's only based on size. So there's our filter, 3 layers. And it looks like this over here. So if we look at it, there's our afferent arteriole, blood comes in here. There's our capillary bed, remember it's in a ball. And then around the capillaries, here's the capillary right here. There's our capillary. See the little fenestrations, the little holes right there. And then there's a basement membrane around that. And then here's our podocyte. And there's our pedicels. And there, the little squiggly red line, that's the filtration slit. So blood's going to come in here. What can go through the filter will. What cannot go through the filter is going to go out this way. So blood comes in. What can go through the filter will. What cannot will go out. Does it make sense? Any questions? Well, remember, in order to make something move, we're going to have a force. And we figured out before, when we looked at regular capillaries, remember regular capillaries look like this. And we have this arteriole, and then on this end, remember, we had a net filtration pressure of 10. But on the other end, we had a net filtration pressure of -8. So across this capillary, our net filtration pressure was 2. And remember, that pressure pushed out 3.5 liters of fluid per day. But here, look how much bigger the arteriole is. And remember, this over here is a venule, but this is another arteriole. It's the only place in your whole body where we have an arteriole, capillaries, and another arteriole. Remember, arterioles can do vasoconstriction and vasodilation, So if I look in here, the pressure is much higher. And if I figure out the net filtration pressure here, it's not 2, it's 10. So instead of getting 3.5 liters pushed out per day, I get 180 liters pushed out per day. To make sense? Any questions? We're going to use basically the exact same equation. The only thing that gets changed is this, but it's exactly the same as interstitial fluid hydrostatic pressure. But C stands for capsule or capsular. So it's the pressure inside the capsule. But look, when I do it, I get 10. And 10 is going to give me 180 liters of fluid per day. So that means that 180 liters are going to come out of the blood into these little tubes. And if I don't get it back out, it's going to become urine. Any questions? So we're going to wind up with a bunch of filtrate. Well, if I take 180 liters, because that's how much comes out in a whole day, and I divide it by 24, that's how much is going to come out in an hour, and then I divide that by 60, and I can figure out how much is going to come out every single minute. And so that's what we're going to look at, is how much is coming out every single minute. And there's a name for that. It's called the GFR. GFR stands for the glomerular filtration rate. And all that is the rate at which we're making filtrate. The rate at that fluid is moving from the blood into the capsule. And if I again take 180 and divide it by 24 and then divide that by 60, I'm going to get this number right here, 125 milliliters per minute. So that's a normal or average GFR. GFR, amount of filtrate formed per minute. And the average is 125 milliliters per minute if I'm filtering 180 liters a day. And this is the ideal GFR. Does everybody understand what GFR is? Again, all it is the rate at which fluid moves from the blood into the filtrate. How fast it happens. Any questions about it? Well, remember this GFR, the ideal is 125 milliliters per minute. You need to remember that number, 125. If we get outside of this 125, we wind up with problems. If you raise the number, if GFR is too high, remember what's going to happen is not only is bad stuff being filtered, but good stuff is being filtered. And if you want to keep it, you have to reabsorb it. But if this number is too high, the fluid passes by so quickly, it can't be reabsorbed. In other words, it's flowing by. And here I am on the side of the river or side of a creek. And I'm trying to grab something. So here it comes floating by. It's something I want to keep. Well, if it's flowing pretty slowly, I can just grab it and get it out. But if it speeds up like crazy, if you have a huge rainstorm and water starts flowing through there really, really fast, this thing's going to zoom by so fast that I won't be able to catch it. And it's going to be lost. It's going to wind up in the urine. So we don't want GFR too high. We also, though, don't want GFR too low, because what happens here is everything gets reabsorbed, and not just the good stuff. Also, waste products get reabsorbed. And so instead of them being excreted, they stay in the body. They're not excreted. They stay in the body. And if they stay in the body, they act like poisons or toxins. And so we sure don't want GFR to be too low either. Does it make sense? Any questions? So in other words, we need to keep GFR right or as close to as ideal as possible. Do you remember what ideal is? What is ideal? 125 milliliters per minute. Again, you don't want to forget that number because that's the number we're trying to keep it at. We don't want it too high. We don't want it too low. We want to keep it right at or as close to 125 milliliters per minute as possible. And so there are two different ways to keep that at 125. One way is called intrinsic. Intrinsic just means inside the kidney. In other words, the kidney does it itself. So the mechanisms are inside the kidney. And so let's look at this intrinsic. It's sort of automatic. It's auto-regulation. It happens in the kidney. And it's really, really important because if you change GFR, you're going to change all kinds of things. So look, if you just change GFR from 125 to something else, it's going to change a lot. So here's the blood pressure in the kidney, and we're going to get 180 liters per day. Look, that blood pressure at the kidney is 100. But if you just raise the blood pressure by 25%, so we're going to go from 100 to 125, look what happens to GFR. It goes from 180 to 225. 225 liters a day, which translates not into 1.5 liters of urine, but 46 liters of urine. We don't want that to happen. We want to keep it the same. And so as the blood pressure goes up, so does urine volume. And so we want to regulate this blood pressure so we can regulate GFR. Again, this is ideal right here. And in order to do that, we have to have this number right here. That's inside the kidney. But there are also controls outside the kidney. And those are related to the nervous system and the endocrine system. And we'll talk about it as well. But let's talk about how we're going to regulate GFR. So look at this. So this could be something like a bathtub or something. In this case, it's, I don't know, some sort of tank. But look, what if I told you to go to your bathtub and I want you to keep the bathtub at this level right here, this tank at this level right here. But look, there's water flowing into your bathtub, it's coming in the faucet. But the drain is also open, so water's flowing out of the bathtub too. Look, there's water coming in right here, but there's also water going out right there. But I want to keep this at this specific level. Think about this. If I want to fill up my bathtub with water, and I want to keep it at that level, but there's water going in and water going out, what if this volume, this level of water changes? Well, look, I can change or adjust or make adjustments to this level two different ways. I can turn the faucet up or down, but I can also open up the drain or close the drain. In other words, I can make more come out here or less. And I can do the same thing here. I can make more drain out or less. Well, it's not a faucet. Remember, these are arterioles, two of them. Remember, arterioles, when you look at them, remember they have two layers. There's a tunica media and a tunica interna. The tunica media, remember, is the muscle, and it goes in a circle around the blood vessel. And remember, muscle can constrict. We get vasoconstriction, or it can relax, and we get vasodilation. So remember, if this constricts, this is going to happen. I'm only going to have that much space for blood to go through. But if it relaxes, that's going to happen. So look, it's like opening up or closing the faucet. So I can, because it's an arteriole, I can vasoconstrict, or I can vasodilate. But I can do the same thing down here. I can vasoconstrict, or I can vasodilate. Does that make sense to everyone? So here's my intake, and here's my drain. And I want to keep GFR at ideal. So look at this. Tell me what you would do if this happened. Look, I want it to go there, but look, it's down here. GFR is too low. I need to bring it back up to ideal. What are the ways that I can bring it back up to ideal? What could I do? What could I do to this one here? In other words, I need more water to come in here. What could I do to that one? Remember, the only things I can do are vasoconstrict and vasodilate. So what would I do here? Yeah, I would vasodilate the afferent arteriole. So I could do that. But what could I do here? This is the drain, remember? I want the fluid to get higher. So what would I do to the drain here? What would I do to the efferent arteriole? Exactly, I could vasoconstrict the efferent arteriole. Or if I really, really wanted it to happen fast, I could do both. And that's exactly how it works. So if GFR is too low, if GFR is too low, I need to increase GFR. And the ways I can do that is by vasodilating the afferent arteriole, vasoconstricting the efferent arteriole, or I could do both. But now look at this one. Here's the level I want it to be at. And it's way up here. GFR is too high. So what can I do to fix this one? Remember, this is the intake. This is where the fluid is coming in. And this is the exit. That's where it's going out. But I want it to go this way. It's too high. So what can I do? I can vasodilate the efferent. That's exactly right. What else can I do? Vasoconstrict the afferent. Exactly. Or if I really, really wanted it to happen quickly, and I could do both. And that's exactly what we see here. So look, GFR is too high. Too high. And so I need to decrease GFR. I need to make it go down. And so I can vasodilate the afferent arteriole, vasoconstrict the afferent arteriole, or I can do both. Does that make sense to everyone? And that's exactly how we're going to regulate GFR. So what we're going to do is change the diameter of this, or change the diameter of that, or maybe both. If I open this up, look, more fluid is going to come in, more pressure, and more is going to get pushed across. If I close this one, less can get out. And so more is going to get pushed across. And then, if I have the same token, if I close this one off, less fluid is going to come in. There's less pressure, and so less is going to be pushed across. Or if I open this up, it's easier for fluid to get out, and so less is pushed across. That's exactly how we're going to change GFR. So look at this. I want you to try to figure these out. So what I want you to do is try to determine what's going to happen. Figure out what's going to happen to blood pressure in here. What's going to happen to net filtration pressure? Remember, that's the number, the 10. And then what's going to happen to GFR? So if we look at the first one, look at A. What did I do here? What I did was I vasoconstricted the afferent arteriole. So what do you think is going to happen to blood pressure in here? Is there going to be more blood or less blood coming in here? If there's less, what's going to happen to blood pressure? It's going to go down. If blood pressure goes down, what's going to happen to net filtration pressure? Remember, blood pressure is the one pushing it out. And if that number gets smaller, what's going to happen to this number? It's going to go down. And if this number goes down, what's going to happen to GFR? It's going to go down too. So if I vasoconstrict the afferent arteriole, this is what's going to happen. Look at this one. Same thing. But here, I dilated. So what happens if I dilate this afferent arteriole? What's going to happen to blood pressure? Is there more blood or less blood going to go in there? So what's going to happen to blood pressure? It's going to go up. If blood pressure goes up, what's going to happen to net filtration pressure? It's going to go up. And if net filtration pressure goes up, what's going to happen to GFR? It's going to go up. And then look at this one, though. I didn't change this one now. What I'm changing is here. I've dilated the efferent arteriole. So the same amount of blood is going to come in, but is it easier to get out or harder to get out? It's easier. So blood's going to flow out really, really easy. So what's going to happen to blood pressure in here? What's going to happen to the amount of blood in here? It's going to go down. If blood pressure goes down, what's going to happen to net filtration pressure? It's going to go down. If net filtration pressure, what happens to GFR? It's going to go down. And last one, look what happens if I dilate this one. So if I dilate this one, I'm going to increase blood pressure. I'm going to increase net filtration pressure and increase GFR. Does it make sense? Everybody figure all these out? So here, I would decrease GFR. Here I would increase GFR. Here I would decrease GFR. Here I would increase GFR. Any questions? Does it make sense? So how do I know what GFR is anyway? I know I can change it, but remember, I want to keep it at 125. So I need to know if it's 125 because I need to know if I need to do this or this or this or this. I have to know what it is. There has to be some sort of sensor telling me what it is. Well, the sensor is actually found in the macula densa cells. So remember, the macula densa cells are in the distal convoluted tubule. So if we just straighten out our nephron, it would look something like this. Just to straighten it out. Here's my proximal convoluted tubule. Here's my loop of Henle. And here's my distal convoluted tubule right here. And remember, at the distal convoluted tubule, I have these special cells. They're tall and they're densely packed. And what they are, remember, are chemoreceptors. They detect a chemical. Anybody remember what chemical they detect? They detect sodium and chloride. And so what they do basically is they count sodiums and chlorides. So as the fluid moves through here, I'm going to start taking out sodiums and taking out chlorides. They're little carriers that pull it out. So let's say we have this many carriers, 5. Well, think about it. 5 carriers can basically pull out 5 sodiums. So if I have 10 sodiums come in and 5 is being pulled out, how many is going to be over here when these are going to count it? 10 came in, 5 was pulled out, there's only 5 left, right? Everybody makes sense with that? What if it wasn't 10 though? I can still only pull out 5, but what if 20 came in? Take out 5, this would be 15. Does that make sense? Does everybody see how that works? Well, Remember, it's based on how fast it comes in, the glomerular filtration rate. So if it's coming in very slow, these might actually have time to take out 5, and then they're still slow. They might have time to take out 15. This would only be 5 now. So look, even though 20 came in when I got over here, there's only 5 left. But what if this fluid is moving really, really, really fast? They might not even have time to do 5. They might only have time to do 3. This number would go up to 17. But if it's going really, really, really slow, slow. So I got that one out, and I've got time to grab another one. And I got that one, and I got time to grab another one. This number is going to go down. Does it make sense? So it's kind of like a little conveyor belt, sort of. So if I wanted to figure out how fast this conveyor belt was going, and I know that every minute 20 of these boxes get removed, and if I sat over here and counted how many did not get removed, I could figure out how fast this was going. Look, 20 are removed, 5 is, this is going at 25. Does that make sense? But what if it wasn't 5 that escaped? What if there were more boxes that escaped? In other words, there were 10 boxes that escaped. I'm still only removing 20 per minute. How fast is this thing going now? It's not going 25. How fast is it going? It's going 30. 20 plus 10 is 30. But what if it wasn't 5 that were escaping? What if only 3 escaped? I'm still removing 20 per minute. But now how fast is my conveyor belt going? 23, that's right. One more time. What if it's not 5? Again, what if it's 7 that are escaping? I'm still going at 20 per minute, but now how fast is my conveyor belt going? It's not 5 over here, 7 per minute. So how fast is this thing going? "27, that's right. So look, I don't need to know exactly how fast this is. I can figure it out if I know how many are being removed and how many are escaping. So what if it's not boxes? What if it's sodiums? So this is a sodium, this is a sodium, this is a sodium, this is a sodium, and so on. These are all sodiums. Well, they're going through that tube. And I have carriers, but the carriers can only remove 20 sodiums per minute. Well, if this thing is going through here at a certain speed, 20, and I'm over here, only 20 are removed, but there's 5 that are escaping. How fast is this fluid moving through this tube? It's moving through at 25 per minute. Does that make sense? So in other words, I can figure out how fast this is going by just counting how many escape, because this number doesn't change. So if this number is 3, that's 20. that rate is 23 per minute. If this number is 5, it's 25 per minute. If it's 7, it's 27 per minute. If it's 10, it's 30 per minute. So look, the faster it goes, the more escape. The faster it goes, the more that escape. So if I count how many escape, I can figure out how fast it goes. And that's exactly what these macula densa cells do. Oops, too far. They count sodiums and chlorides. These macula densa cells, they count sodiums and chlorides. And the more sodium that goes by, the higher the GFR. The fewer sodiums that are down here, the lower the GFR. This is a GIF, and you can see it if you look at the PowerPoint. But basically, it's the same thing. These carriers remove the sodiums, and it's at a set rate. So if I know what that rate is, it's 20, and I know how many escape, I can figure out the speed at which it's going. I can figure out the GFR. It's kind of like this, and if you watch this, you might get a kick out of it. But anyway, it's sort of like this. Anybody ever seen the Lucy show where this episode where she and Ethel are wrapping candy with papers? And so look, this thing right now is going very slow. So if I were to be over here in the room and I were to count how many arrive unwrapped, it would probably be a very small number because it's easy to wrap these things. It's going through. I can easily pull off the sodiums. So everybody's happy, pull it off, nothing. Look, nothing's escaping over here. The number is really low. But what happens when you speed up GFR? If you speed up GFR, they can't wrap or they can't pull out any sodiums any faster. So sodiums are going to escape. And so if I count how many sodiums escape, I can figure out how fast this conveyor belt is going. I can figure out GFR. And that's exactly what happens. Make sense to everyone? So what those macula densa cells do is they monitor GFR, the distal convoluted tubule. So if more sodiums are making it all the way through the distal convoluted tubule, that means that GFR is fast. So if too many sodiums pass by, GFR is too fast. And what they're going to do is they're going to do something to slow down GFR. What they do is they release hormones. Remember, if GFR is too high, what are we going to do to fix it? We're either going to vasoconstrict the afferent arteriole or vasodilate the efferent arteriole, or both. So it's too high. So what the macula densa cells do is they release these hormones, and look what happens. We get vasoconstriction of the afferent arteriole. We're going to get this. And if that happens, GFR is going to go down. Make sense? This is what we're going to do. Any questions? So that is if there's too much sodium going by. But look, what happens if there's too little sodium going by? That means that those carriers pulled out all the sodium. So very few of them pass by these macula densa cells. So that means GFR is too low. And so what they're going to do is they're going to release a different hormone. That hormone is going to cause renin to be released. Renin is an enzyme. What that enzyme does is it triggers a cascade. It triggers a cascade. And so renin converts something that's already in the blood. It's made by the liver. It's called angiotensinogen. Renin converts it into angiotensin 1. Look at this word. We already talked about it when we talked about endocrine system. Angio means artery, and tensin, like tension, means to constrict. So it's a vasoconstrictor. This one's not a very potent vasoconstrictor, though. But when it circulates, it comes across this enzyme. This is found in the lungs. It's found in some other places, but primarily in the lungs. And it converts it into angiotensin 2, which is a very, very active vasoconstrictor. And that causes the efferent arteriole to constrict. And so this is what happens. And so GFR goes up. Make sense? Any questions? Another thing this hormone does is it increases the absorption of sodium and causes ADH to be released. Remember, if the kidney absorbs more sodium, water is going to come with it. And ADH, remember, lowers urine volume. And all those things are going to increase blood volume. And if they increase blood volume, they're going to increase blood pressure. And if you increase blood pressure, remember you're going to increase in net filtration pressure. And if that goes up, GFR is going to go up. So this hormone is going to work two different ways. It's going to cause a constriction, but it's also going to increase blood volume and increase blood pressure and increase net filtration pressure. And that also causes GFR to go up. And it looks like this. Okay, that's what happens inside the kidney. But we can also have stuff outside the kidney, which increases or decreases GFR. Primarily, those are the hormones we've already talked about, epi and norepi. And epi and norepi are going to cause afferent arterioles to constrict. So we're going to vasoconstriction of the afferent arteriole. What is that going to do to GFR? It's going to decrease GFR. In other words, this is going to happen. So what's going to happen is we're going to wind up with a decrease in GFR. Any questions? This doesn't have anything to do with this, but I want to also point out something. Do you remember this enzyme right here? We just said converted this to this. This enzyme is actually how COVID gets into your cells. So any cell that has this enzyme on its surface, that coronavirus can get into it. And so there's a lot of it in the lungs. It's also found in lots of other places. And so if you've been keeping up with the whole COVID thing, first people said that just the lungs were involved. But now they're finding out lots of other organs are also involved. Any organ expressing this enzyme right here, and usually it's called ACE, but it's angiotensin converting enzyme. Any cell that's expressing that can be infected by that virus. Any questions? So, a couple of things. Go back if you didn't get it, and make sure you understand this. So you should be able to determine what's going to happen to each one of those if any of these things happen. And then remember that we want to adjust GFR. So we can either make it go up or we can make it go down. But in order to do that, we have to know what the rate already is. And so we have to figure out the rate. And that is done with these macula densa cells. And they do it by monitoring sodium and chloride. So the sodium and the chloride is removed at a set rate. And what they do is they sit over here and they count how many escape. And so by counting how many they escape and knowing how many removed, you can figure out GFR. It's the same thing here with the Lucy and Ethel. If we know how many they can wrap, they can take off, and we know over here how many show up that they didn't wrap, we can figure out this rate. And here, look, the rate is very slow because this number that escapes is very low. But if we look here, look, the rate is very fast. That thing's zooming along because they can still only take off the same number, but they're escaping. And if I know how many they can wrap and how many escape, then I can figure out how fast this thing is moving, the GFR. And then I want to adjust the GFR. So if it's too low, I need to do something to make it go back up. And so I do vasoconstriction of the afferent arteriole. And that, sorry, let's go back. If it's too high, I need to make it go down. I need to make it go down. And so I'm going to do something to vasoconstrict the afferent arteriole, which is going to decrease GFR. If it's too low, I need to do something to make it go back up. And so I'm going to go through this pathway, and this is going to happen. I'm going to vasoconstrict here. And if I vasoconstrict there, more is going to go this way. I'm going to increase GFR. Any questions? But again, make sure that you understand the ways you can increase or decrease GFR. This is a good summary slide right here. So go and look at it until you're able to answer all these questions. And then here are the answers. So it's sort of a little self-quiz. Okay, so that's as far as I had planned to go today. Anybody have any questions? So before we meet next time, which should be tomorrow, make sure you go through and look over the case studies so that if you have questions, I can answer them. And once you figure out which case studies you're going to do, then you can go ahead and get started on those. But remember, you only have to do 3. So you have 2 to choose from for exam 1, 2 to choose from exam 2, 2 to choose from exam 3. Any questions? Okay, so I'm going to go ahead and stop recording.